Hello Year 12, welcome to another flip video for maintaining a balance. Let's just jump in. So, we are looking at an experiment today, and what's interesting about this particular experiment is that you don't have to plan it or anything like that. You basically just have to perform it. You have to perform an experiment so that it demonstrates the effect of dissolved carbon dioxide on the pH of water. Now we know that what the pH of water is, so basically we just need to figure out what the hell does carbon dioxide do to it. Alright, let's get stuck into it. So, firstly, in order to do this experiment we need to find a source of carbon dioxide. Now, you and I are probably very familiar with the idea that we exhale carbon dioxide, which is great. That gives us, a ready supply of it. But we can also get carbon dioxide from a reaction with vinegar and bicarbonate soda. And what we're going to do is we're going to put these sources of carbon dioxide through the lime water test, just to verify that in fact we do have carbon dioxide there. What will happen is that carbon dioxide will turn a clear liquid that is called lime water. Well, it doesn't, not like lime the fruit, but lime water with the it's called lime water, and a white precipitate will form when exposed to carbon dioxide. So it'll be a clear liquid, and then little white particles will form if it is exposed to carbon dioxide. So let's have a look at what that is. Essentially, we could put we could get a syringe of carbon dioxide and put it into lime water, and it would go clear. And then, just to verify that our breath does in fact have carbon dioxide in it, we would breathe through the straw, and if we know that that is carbon dioxide over here in the syringe, then these two will be the same positive test. You'll get that precipitate forming. Alright, so another option is looking at the setup of a conical flask with a stopper and a glass tube and lime water with just water in it, and then doing the same reaction with vinegar and baking soda. And we know that baking soda releases carbon dioxide from here, and so we will have a validation of our experiment, the fact that water did not produce the positive result in lime water, but the reaction of vinegar and baking soda did. So that's what our experiment's going to look like. All you have to do is perform it and record some results and tell me what's going on. So let's have a look. This is our method. We add a universal indicator to 100 ml of water in a beaker. We are going to use our color indicator chart to record the pH. We should already know what our expected pH and color should be, but whatever it is, you record it regardless. And then you are going to exhale through a straw into the beaker of water for 1 minute. So, safety first. Don't suck up the straw. You're blowing out. You're exhaling. You're not inhaling. Alright, so we don't want you to consume any of the liquid in the beaker. And you also don't want to it's not like you're trying to blow the world's biggest bubbles, alright? You've got a beaker of liquid with the universal indicator in it. You just want to blow steady stream of bubbles, right? Making sure that the gas from your breath is circulating through that beaker of water. Alright, after a minute you are going to use that color indicator chart again to record the pH. It is a very, very straightforward experiment. It is a very quick experiment, so we will not be spending too much time holding your hand doing this. It is a very, very simple experiment. So, once we've done that, we need to start asking some questions. So, the first one is the obvious. What happened to the pH? Did it go up? Did it go down? What's that? What is that? Then we need to look at the an inference. What might you be able to make about the effect of having too much carbon dioxide in blood? Well, we know plasma is mostly water, and so if you are dissolving, we've looked at one of our previous dot point videos was about how is carbon dioxide transported in the blood. Well, one of them is dissolved, and and so we want to know about what happens if you've got too much dissolved carbon dioxide in the blood. That's a pretty important question. Particularly because it's related to pH, and we know we've done an experiment on pH when we related it to enzyme function. So I think you guys kind of get where we're going with this one, but I think asking these questions directly is going to be a bit of a discussion in the lab. So it's about kind of going, look, one dot point can relate to many others. And this experiment gives you first-hand information about the importance of regulating this particular substance called carbon dioxide. Alright, that was a quick and simple one. See you in school.